Hello, everyone. My name is Maliha. I am a biomedical research intern here at Berkeley Pharmatech and an undergraduate student at UC Santa Barbara. Today, I will be discussing a topic with you under the field of endocrinology and women's health, more specifically, the effects of exogenous sex hormones on the female human brain. I would like to start off by giving you a brief overview of what we will be covering today. So I'm going to begin with the large gap in biomedical research for women's health, and I will give you a little bit of history on why this is. We will talk about the possible use of exogenous hormone therapy to delay the onset of Alzheimer's disease, and this is a really current topic in research today. Another very current topic in research today is the unknown effects of the oral contraceptives on the human brain. I will then be covering a study done at UC Santa Barbara about the effects of progesterone suppression on the medial temporal lobe morphology. I will then proceed with further research questions from these studies, and I will conclude with the need for personalized medicine for women. I'd like to start off with talking about the large gap in biomedical research for women's health. In 1992, the FDA reported that women were generally underrepresented in drug trials, and even when women were included in large numbers, data were not analyzed to determine sex-related differences in drug responses. And why has decades of biomedical research been centered around the male model? Well, between the 1970s and 1990s, these regulatory organizations had policies that took women who were able to bear children out of these studies because they have a finite number of eggs and drugs could be toxic to these eggs. This opened up a huge bias towards women. Additionally, another reason that women were taken out of these trials was because of a myth that said that women were more variable than men because they have a menstrual cycle, and within a menstrual cycle, women have fluctuating hormone concentrations. And in order to get a more pure study with less variable results, men were used because they were seemingly less variable. This proved to be myth and an assumption on bias. In a 2010 study, it was concluded that the female variability argument had no actual foundation. So what were some of the results of this decade's worth of bias? Well, between 1997 and 2000, 10 prescription drugs were taken off market by the FDA because of severe adverse effects. 8 out of 10 of these effects caused greater health risks in women. A study conducted in 2018 explained that this is due to serious male biases in basic, preclinical, and clinical research. As quoted before, even when women were included in large numbers, data were not analyzed to determine sex-related differences in drug responses. So instead of working sex differences into research, researchers just completely left women out of the studies. The conflict is that researchers applied these results surrounding the male model to women as well, not even taking into account these sex differences. And because of this, women have suffered the consequences of drugs that weren't really designed for them. One example of this is metabolism. Women generally have a faster metabolism than men, yet this isn't accounted for in research. In 2014, the NIH acknowledged that there is a male bias in preclinical trials. In 2016, the NIH stated that research it granted had to include female animals. So it wasn't until recently that these biases were acknowledged. And really, women have lost decades of research in the biomedical industry because of this. Here are some neurological conditions with sex differences. So autism in boys, multiple sclerosis in women, Parkinson's in men, depression in women, and Alzheimer's in women. So it's clear here that there are sex differences between different disorders and diseases. So even though these differences exist, treatment options do not account for these sex differences between men and women. There is a need for further exploration of the mechanism by which sex differences occur. One possibility for neurological conditions is neuroinflammation, which is a very important contributor to the pathophysiology of neurological disorders. Men and women are known to have differing neuroinflammatory states, which can contribute to the differing prominence of these conditions in men and women. Research is being conducted to find why women have an increased risk of Alzheimer's disease. Estrogen is an extremely important sex steroid hormone that is present in different concentrations throughout a woman's life. To give you a little bit of context about sex steroid hormones and their importance, sex steroid hormones are some of the most impactful, powerful hormones in our body because they contribute to our prenatal development, pubertal development, menopausal, and aging. Sex steroid hormones contribute to behavior, physiology, and functionality of humans all around. So again, estrogen is an extremely important sex steroid hormone that is present in different concentrations throughout a woman's life, and it regulates the chemical that allows for communication among cells. During the developmental transition of menopause, estrogen levels decline significantly, contributing to lower brain cell energy. So the big research question is it possible that this decline in estrogen levels in women undergoing menopause is a contributing factor to the cognitive decline and larger risk of Alzheimer's disease in women? Well, research suggests that the timing of hormone therapy among women who undergo menopause can help reduce the risk of Alzheimer's disease and maintain cognitive function in women. The timing of hormone therapy is being studied to understand the right timing for optimal treatment. Hormonal replacement therapy later in life could have negative effects on cognition, but treatment at the beginning of the menopausal transition could actually have beneficial effects. A recent study showed that women who took estrogen after the age of 65 were more likely to struggle with thinking and memory, whereas women who started taking estrogen between the ages of 50 and 54 were not. These are some really promising results, and this research is really being done currently in this field to figure out whether this could be a possible prevention mechanism for the onset of Alzheimer's disease. Shifting gears a little bit to the oral contraceptive. This was historical. Women had rights over reproducibility. This allowed them to get careers and education. Currently, it's used by 100 million women. But we do not know the effects that the chronic sex steroid hormone suppression from the oral contraceptive has on the central nervous system. The hippocampus and prefrontal cortex contain many estrogen receptors, and we do not know the effects of chronic suppression of hormones like estrogen or progesterone has on these areas. So really, the oral contraceptive has been great in fertility and reproductive health, but we don't really have enough research on how it affects our brain. And this is not me saying go off the pill, but this is me saying we need to have more research on the effects of this pill, because there is not enough research being done, and we owe it to women to have better research on this oral contraceptive because it's used by so many. So the mechanism behind the oral contraceptive is important in understanding why this may have effects on our central nervous system. So generally, the oral contraceptive is a mixture or one of these two hormones, ethinylestradiol and progestin. These hormones bind with endogenous sex steroid hormone receptors within our bodies. These exogenous hormones limit the hypothalamic production of gonadotropin-releasing hormone, or GnRH, and this inhibits the release of luteinizing hormone and follicle-stimulating hormone by the anterior pituitary. The decrease of FSH stops follicle growth and LH surge that triggers ovulation. So the oral contraceptive is chronically suppressing estradiol and progesterone production. With this suppression of such important sex steroid hormones that are important to our developmental health, there is called a question as what this chronic suppression may do to our brains. So a study done at the Emily Jacobs lab at UC Santa Barbara found that progesterone shapes medial temporal lobe volume across the human menstrual cycle. So in humans and non-human primates, there's a known effect of sex steroid hormones on hippocampal morphology, but we do not know the impact of sex hormone fluctuations on hippocampal morphology in humans. High-resolution imaging was used on the medial temporal lobe across two 30-day periods in order to determine if variation in sex hormone concentrations across the menstrual cycle impacts the medial temporal lobe morphology. So in the first 30-day period, the relation between the medial temporal lobe subregion volume and endogenous fluctuation in sex hormones across a complete menstrual cycle was assessed. In the second 30-day period, the effects of chronic suppression of progesterone was assessed. So in study 1, the endogenous fluctuations in progesterone through the menstrual cycle corresponded with changes in gray matter volume in multiple regions within the MTL. This included CA2-3, parohippocampal, entorhinal, and perirhinal cortex. In study 2, this chronic suppression of progesterone abolished these volumetric fluctuations that were found in study 1. These results indicate that progesterone shapes medial temporal volume across the human menstrual cycle, and chronic suppression of this sex hormone diminishes these volumetric changes. This is fundamental to understanding the brain morphology throughout the human menstrual cycle. And future studies could research whether progesterone-mediated changes to the MTL have correlations with mood, cognition, navigation, and decision-making. This is also significant for adding research to the role of sex hormones in neuropsychiatric and neurological disorders. So further research questions from this study. If progesterone suppression using oral contraceptive inhibits volumetric changes in the MTL, could this have long-term or short-term impacts in areas such as behavior or brain morphology for women who take oral contraceptives? And could this inhibition in changes to medial temporal lobe volume have any effects on women who begin oral contraceptives earlier on in development? Because oral contraceptive is given to girls starting at a very early developmental age during the prepubertal period. Age 10 to 17 years old are prescribed the oral contraceptive for whatever reason. And in the US, 36% of 13 to 18-year-olds fill the OC prescription. So more questions on the oral contraceptive could be, can this chronic suppression of such an important sex hormone, such as progesterone, have an effect on the human brain over evolutionary time and populations in the next decades? And in what ways could oral contraceptives alter our future in endocrinology? Additionally, can oral contraceptive given at early developmental stages possibly alter organizational effects of endogenous sex hormones because of its chronic suppression of the ovarian hormone at such an early developmental stage? At developmental stages, such as prepubertal, we have large fluctuations in hormone concentrations. So can the suppression of these hormones lead to some effects in possibly behavior or function? Or development of girls who are given the oral contraceptive at ages like 10 years old. So I'd like to discuss a little bit about improving oral contraceptive research design because the studies that are out right now are generally poorly executed and seemingly inconclusive. Within the studies, they don't really take into account the type of oral contraceptive brand that is taken by subjects because brand can alter effects. So the oral contraceptive brand could alter effects based on different concentrations and amounts of the hormones. You know, the amount of time on the oral contraceptive, whether a woman has been on it for 2 years, 10 years, 20 years. This is a really large variable within studies for the oral contraceptive and past usage of the oral contraceptive. It's not enough to say that a woman in a possible control group is not on the oral contraceptive because what if she was on it for the past 20 years? So there's just a few recommendations for better study design on the oral contraceptive, but there are many more. I'd like to conclude with discussing the need for personalized health for women. Not only do women need medicine designed and taking into account these sex differences, but more personalized medicine in terms of biology, genetics, health history, and demographics can more efficiently help women in treatment. This could be in technologies including genetic sequencing, for example. Here are my citations. I'd like to conclude by saying thank you so much for listening to my presentation. I hope that you found useful information from this topic. Women's health is a largely overlooked field, and it's important to be giving it the attention that it needs. Thank you.